Stem cell biology is actually very, very simple. You all know that when a cell divides, it has two daughter cells. The one difference between a blood-forming stem cell at the top and the daughter cells that will eventually go on to make blood is that one of the two daughter cells, on average, is still a stem cell. For the scientists in the audience, it's the same gene expression program. That's it. That's the difference between a stem cell and any other cell in a tissue or organ. We found years ago that when we could isolate and follow the behavior of, say, the blood-forming stem cell, that it was the only cell that could regenerate the blood-forming system in a bone marrow transplant. And we found, in fact, as you'll see, that quite often all the other cells in the bone marrow transplant don't help you, with a few exceptions, and that it would be cleaner and safer and better to purify the cells to transplant it. Blood-forming stem cells, as I show in red, make blood and only blood. Brain-forming stem cells, as my wife Ann and I discovered, make brain and only brain. There are fraudulent clinics and fraudulent claims everywhere in the press because they want to take advantage of the word stem cell and then be able to sell you a treatment not tested under IRB, safe conditions, not even brought forward or tested by the FDA. The reason I'm saying it right now before I forget it is I gave a talk like this in Great Falls, Montana, to people from Great Falls. And at the end of my talk, 2 of the 180 people came down and said they had spent the rest of their life savings for fraudulent stem cell therapies for their spouses. They're willing to take your money, but you must, as caregivers, as patients, as future patients, always ask 2 questions. Who is the name of the person who protected the patient during the clinical trial, head of the IRB it's called, and what's the number of the FDA protocol that approved it as being safe and effective? My ad's over now, but I just wanted to make sure everybody understood it because this is taking the hopes and dreams of people and the money they had left for the rest of their lives for profitable income. It's almost impossible to understand. So when we isolated the blood-forming stem cell, we wanted right away to do what Robert Koch and Louis Pasteur and all of those people did, to see if we could improve therapies by using them. So the upper left would be a blood-forming tissue like bone marrow. Now, there's a way that docs have made to bring cells out of the bone marrow that are early stage development. It's called mobilization. And so either bone marrow or mobilized peripheral blood, MPB for the science, could be a source of blood-forming stem cells. We found that in patients who had metastatic breast cancer or hard-to-treat non-Hodgkin's lymphoma or acute leukemia or multiple myeloma, that the bone marrow and the mobilized blood had cancer cells in it. And when we wanted to move to trials with highly purified cells, I learned something about education that has been very disturbing to me ever since. I would go to the breast cancer doctors and say, instead of giving patients a very high dose of chemotherapy, a lethal dose, and then rescuing them with cancer-contaminated bone marrow or mobilized blood, wouldn't it be better to treat them with cancer-free blood-forming stem cells? And whether they were bone marrow transplanters or breast cancer doctors, they say, oh, it's been proven it doesn't work. And how would you know that if cancer cells put it back in the body would grow? And it was that moment of frustration that I remember as a happy pre-med here at Montana State, that at places I had gone before and places I've encountered since, pre-meds are the most disciplined memorizers of anything. And it's a horror even to know you're competing with them to do your life work, that is, get into medical school. But never in their career, for almost all of them, have they learned how the information was obtained that they memorized from textbooks or pearls of wisdom from their clinical colleagues. And what happens at Stanford, and what happens, I remember, at Montana State, is that you get in the lab while you're doing your undergraduate work, and you do experiments, and you learn how hard, but how wonderful it is to dig things out of nature and find out the truth. You're then prepared to listen to somebody like me who says, don't give them that thing up on the left, separate the cells for pure stem cells, and when we did, they were 250,000-fold depleted of the contaminating cancer cells. In fact, we did a clinical trial that I'm going to show you next, where we gave high doses of 3 drugs, each of which kill a fraction of the cancer cells. And when women come in with breast cancer that's observable, or leukemia patients come in, they usually have about 100 billion cancer cells in their body. And so that's the task. Every last cancer cell has to be treated. And the doses of the drugs that were mainly being used cured nobody, because they got to the point that they had blood formation failure because you killed off all the blood-forming cells. So in order to be able to rescue them, people first harvested their bone marrow or their mobilized blood, froze it down, gave them those 3 drugs at the top, and then at the end they thawed the tissue and infused it in and gave cancer in the woman at the top. So she had cancer cells in the inoculation. The one at the bottom got cancer-free stem cells. And this isn't about single women. This is about a clinical trial we did at Stanford, funded by a company I had started and which later got bought, and I'll tell you about that also. This kind of plot is overall survival versus the time after the treatment. And what you could see, the women who got cancer-contaminated rescue, half of them were dead, that's the 50% point, at 26 months. That's exactly the same before that therapy. That's exactly the same, unfortunately, today with metastatic breast cancer. Half dead, 26 months, almost all dead by about 11 or 12 years. The blue line is the patients who got cancer-free stem cells. Half of them dead at 10 years instead of 2 years. So they had 8 years of life with their families, their companies, whatever. And 1/3 of them are alive without disease today, 21 to 23 years after that clinical trial. So you must be thinking, why did Irv say we have nothing better today? That's better. Of the 50,000 women in the US who reach the stage of metastatic breast cancer, you have a chance, if this extrapolates to large numbers, to cure 1/3 of them, or maybe around 16,000 to 17,000 women every year in the US alone. So it was that kind of promise of stem cells and the promise of some of the other things I'll show you that come from stem cells, that led a very large pharmaceutical company to say, you cannot afford to do all the trials you need to do. You can't move into the fields you want to. And we knew that part was true. There was just no way you could finance these large clinical trials. They said, let us help you. We will do everything you want going forward. And so it was sold. And I was still an advisor for a few years. But the function of a company is to make a profit. And I want to say this in the clearest sense, not antagonizing that, not saying that's wrong. That's how it works in any society that I know. The principal function of a company is to make a profit. And the other thing I didn't know is that the leadership in companies changes about every 5 years. So that promise to carry it forward, to carry it into all diseases, never happened. And when I talked to the current bosses of the company, they didn't even know they were involved with us. So we have this issue that we developed a therapy that could cure 1/3 and extend the lifespan dramatically of the others. And I'll come back to that point, except I want you to know that that exists. Now, in calculating the number of cancer cells that we found in the initial mobilized blood, that the patients who got cancer-free stem cells were there, I estimate somewhere between 10 to 100,000 of cancer cells were left in their body. But they were wherever they were. No surgery could get them. No radiotherapy could find them. You got to give systemic therapy. But at the end of that period, when we transplanted them with cancer-free stem cells, instead of having 100 billion cancer cells, they may have had 10,000. And that's what new therapies added on to this can and will do somehow. I negotiated from that company to get back all of our intellectual property and the rights to make the antibody and put it in a not-for-profit setting at Stanford Hospital and Stanford Medicine. So we'll see if we can convince the next generation of former pre-meds that it might be worth this for their patients rather than what's available. Here is the most difficult slide on earth, right? I want you all to read and memorize all the little stuff that's next to it. This represents about 15 years of work in my lab. At the very tip top is that blood-forming stem cell. We now know it's 1 in 100,000 cells in the bone marrow. And the very first step it takes down to the cell with a blue box next to it, that single cell that's downstream, one step, can make all the blood cells just like a stem cell, but it can't self-renew. So it runs out of the ability to regenerate itself within 12 weeks. So the very first thing, if you're a scientist, is to say, and I won't tell you because I don't know the answer, is what's different between the stem cell that makes blood and self-renews and the next cell down that doesn't. And by working it out, we could find the identity of every decision-making cell between the blood-forming stem cell at the top and blood at the bottom. Because of the advance in studying the genome and the what's called RNA sequences, we also took those purified cells, and we know a lot about the potential of what can happen. So it's a big and important thing. But I wanted to find out about human leukemia. Oops. So in the late 1900s, I had 2 postdocs from Japan, Toshi Miyamoto and Koichi Akashi. And they said, Irv, you know that at Hiroshima Hospital, with foresight, the doctors who were treating patients who got leukemia after the 1945 bomb, with increased incidences peaking around 10 to 12 years later, they saved the bone marrows as living cells from those patients at diagnosis and at several times thereafter. And nobody had that forethought in the US so that you could go back and study the disease. So we did. The upper left was that top cell, the real stem cell. So we called it LT for long term. The top right was the cell that couldn't on its own self-renew, but could make all blood cells. When we analyzed those samples from Hiroshima Hospital, both by tests in the test tube, but also because we could transfer the cells into an immune-deficient mouse, which we call a SCID-HU mouse, severe combined immune deficient. One of the leaders in that field right here is Kim Hasenkrug now, and we can talk about that in a bit. We found that the leukemia cell, that was the leukemia stem cell, was at the stage of that non-self-renewing cell. And the chromosomal event that led to it was in every single cell. So it came from a single cell that had a chromosomal event that led to the leukemia. So that was important. We could transfer the leukemia. We knew it was the stem cell. If we let it make the daughter cells below it on that tree, they didn't have the leukemia. They had all the same mutations, but they didn't have the other genes expressed that could allow it to be a leukemia. So this was a big change in how you think about leukemia. But the most important finding is on the left. 5 to 40% of the highly purified blood-forming stem cells from those Hiroshima Hospital patients who already had leukemia had the same chromosomal event. So it says AML1/ETO. That's a chromosomal translocation. And the DNA sequence is a marker of that cell and all of its daughter cells. And it brings in a gene called AML that plays some role in leukemia. But you can see from that, the blood-forming stem cells that had only that first step only make normal blood. So it wasn't enough to make a leukemia. Lots of people saw this, but hardly anybody thought, what does it mean? So now you have a clonal marker, we call it, a translocation, a DNA change that we can mark it. And we know it's the exact daughter of the leukemia stem cells that cause the disease that kills the patient. But the first step wasn't enough. More had to happen. And that really changed our thinking, at least, and hopefully a couple of other people's thinking about the disease. So this is a diagram. If you look at the left going across the top, so instead of stem cell on the top, blood on the bottom, it's stem cell on the left, blood on the right. And each of those steps now with simpler names, hardly any Latin or Greek, says, this is how you make blood. The mutation that we saw, number 1, AML1-ETO, was that chromosomal change. That happened at the same rate in all of the cells in the blood-forming system. But the only one that would keep the cell alive by self-renewal and therefore be able to build daughter cells and daughter cells and daughter cells was the stem cell. We went on later to show if we put that particular chromosome change into a cell that was further downstream, further to the right, it didn't convert them into self-renewing cells. It didn't cause leukemia on its own. So now we knew for sure it had to be more than one event, and we should look for what those events are. So we made a hypothesis that way down in the bottom, 1 through 7, are the changes that that blood-forming stem cell had accumulated over time. So that's 7. And we predicted if we could know all of the mutations in a person's leukemia, we could take blood-forming stem cells one at a time and said, do you have number 1? Do you have number 1 and 2? 1, 2, and 3, and so on. So that's the test that we did. This is for the scientists in the audience, how we did. It's actually, you don't need to have science. So we isolated AML, the leukemia, from patient number 70 at Stanford. Neither I nor you will ever know that person's name. This is a list of the mutations that we found in the whole leukemia. It's a lot. The ones in red are ones that occur over and over again in people who get this leukemia. So we made a DNA test for each one of those, a very simple DNA test. Then we isolated single blood-forming stem cells. I apologize, it says HSC for the Latin hematopoietic stem cell, but just think blood-forming stem cell. And now we interrogated each single cell to ask what happens. And what happened, that's patient 70 on the bottom, is we found the first mutation. So it was a blue cell. We found out something immediately. It was amazing. That mutation occurred in only one blood-forming stem cell in the whole body. But we could stick a needle in any bone marrow and find that cell amongst the blood-forming stem cells. And the total number, that's kind of rough there, of the blood-forming stem cells in the body didn't increase. So that led us to find and publish that there's a home that stem cells live in. And it's got room for one stem cell only. And when a more competitive stem cell migrates from the bone marrow to the blood and back to the bone marrow, it can outcompete that cell. So the increase in that number is at the expense of normal blood-forming stem cells. That gene has a certain function, and all of us have 2 copies of any particular gene for our parental origin chromosomes that came the genes, mother and father. The second mutation in that patient and the next one we did was the other copy, and both of them destroyed that gene. And when we experimentally destroy just that gene in a blood-forming stem cell and transplant it, it outcompetes normal stem cells. And as I'll show you in just a minute, causes a disease, short of leukemia. And the rest, there, it's not important except to say, this is how you could use single-cell genetic analysis to start to understand. And let me be clear that whatever events are happening in the stem cell have the same probability of happening to any cell in the body. And that cell in the body could be a late-stage blood cell, it could be a brain cell, it could be a brain stem cell. And so we need to get this lesson clear, because what happened here in the blood-forming system could easily happen, for example, in the brain-forming system. The only cell that perpetuates the ability to build all the way to the leukemia is a stem cell. So our lesson was, in acute myelogenous leukemia, the process of developing the pre-leukemia starts with mutations in a single cell that make that cell more competitive. Second, in all of our looking, we've never found a stem cell which is accumulated all but at the last mutation, and maybe the last mutation, that is a leukemia at the stage of the stem cell. So that home I mentioned, it's a very restrictive home. You can't expand it beyond the number of cells you have. If it's true for leukemia, we predict, and we're testing, it's pretty sure it's true, that every tissue that has a normal stem cell, and you can develop a cancer in that tissue, is the same. We're studying glioblastoma, and almost always lethal brain cancer right now. because we can get out normal blood-forming stem cell, brain-forming stem cells from the original surgical sample to take it out. So this is really important. And it says, the word clone means all the daughters from a single cell, okay? Don't want to make that too fancy. Clonal competition by precancerous stem cells can lead to diseases caused by the mutations. And we've published that over the years. So I changed the cartoon, or actually my wife changed the cartoon. I'm technically incompetent. So let's work from the bottom up. That one with 7 mutations is a leukemia, and that kills most of the people who get it. The next one up is a disease of elderly people called myelodysplastic syndrome. Don't blame me for the Latin name. What it is somebody my age goes in for a blood test, feels pretty good, and they find out on the blood test, I don't have enough red cells, or I don't have enough clotting platelets, or I don't have enough neutrophils to fight off infection. And what we discovered is that the first chromosomal change that occurs, and a couple more after that, lead to all of the blood stem cells now taken over 60 years of life by that one cell. It's a very good, very strong, very competitive stem cell, except when it tries to make blood, either the red cell precursor or the platelet precursor or the neutrophil precursor is eaten by scavenger macrophages. That's the mechanism of the disease. The frequency of myelodysplastic syndrome is higher than the frequency of acute leukemia. Then a very smart student who had been in my lab at Stanford as an undergraduate and a medical student, Sid Jaiswal, started looking at the bloods of all of our ages, of people who didn't have disease. And he found that expansion of a clone, higher and higher frequency the older you got, 100 to 1,000 times higher than the incidence of leukemia, because it didn't have to go all the way to cause that. So that's now called clonal hematopoiesis. And if you go to the experimental lab and be tested, you'll know that you have a much higher risk of getting the myelodysplastic syndrome or acute leukemia. But it's not a sure thing that it will get there while you're still alive, because the mutations are rare and slow. But those people have the highest risk factor for coronary obstruction, carotid obstruction, stroke, big bellying out of the aorta into an aneurysm, which if it breaks open, you're dead on the spot. And that's even higher incidence. So this is a hidden set of events for almost all docs. But what we're doing by analyzing purified stem cells is finding out the steps on the way. I took a long time on that one, but I want to now give you another example of what you can learn from stem cells. So David Traver and I in 1998, Sid Jaiswal, Ravi Majeti and I in 2000s, compared pure leukemia stem cells and pure normal stem cells and the stage of the cell that comes from them for which genes are expressed high, medium, and low. And the very first gene, this is luck, that we saw made a protein on the leukemia stem cell that told the scavenger cells in the body, don't eat me. And the person who had first discovered that gene thought it was just a way to measure the lifespan of a red blood cell. So it's on in a red blood cell. Red cells don't have nuclei. They don't make new proteins. So what you got at the birth of the red cell is what you got. And of course, as time goes on, it fades away and fades away. The eating cell, called a scavenger macrophage, waits till it's gone away enough and then eats it. And the mechanism is the molecule, the don't eat me signal, which I apologize, is called CD47. Please don't memorize that. Interacting with a receptor on macrophages that paralyzes those macrophages. And I mean paralyzes in the real sense. So normally a macrophage has to engulf a bacteria or a bad cell or a cancer cell, and it uses muscle proteins, myosin and actin, to move itself around and engulf, eat, and kill. But they're paralyzed and can't do it. And we realized right away that if you have a don't eat me signal that came up like that, it probably happened in a cell that in the early stages of development of the leukemia said, eat me. And we had discovered, but weren't thinking right about it, in 1994, that in the lifespan of most blood cells, they turn on programmed cell death, but before they die, they put on an eat me signal, and they die inside of these scavenger macrophages. And for the immunologists in the audience, if the cell dies in its tissue space, what it releases activates macrophages through the TLR system. As Mark Judela will tell you in the next lecture, we have 5 tonight. And that allows that macrophage to decorate the cell with a protein that is an eat me signal. So it spits out the protein. If it sticks, they eat it. Very simple. So now we had the beginnings of a system that we had discovered in leukemia. So we wanted to test it. Mark Chow, Ravi Majeti, and I took purified human bone marrow leukemia stem cells. We painted them green with a dye. We put them together with human eating cells, macrophages. And now, if you see the red, those are antibodies, proteins that we made that could block the interaction between the CD47 on the tumor cell and the receptor on the macrophage that, if it's engaged, paralyzes the macrophage so it can't do it. So we put the antibody in, and on the top right, you could see those human primary leukemia stem cells. Now we're doing real human leukemia. We're not making a mouse model of leukemia. It's real leukemia. And they got eaten. Whereas other antibodies that bind to other entities in the surface can't overcome the don't eat me signal. We could transfer the leukemia into immune deficient mice so we could do a real preclinical test. What you're seeing on the left is the bone marrow. The red is the bone. It's monotonous. There's only one cell type there. It's not making blood. It's leukemia. And we waited till this stage, which is a late stage in human leukemia. And we put in our blocking antibody, and 90% of them were cured. 10%, where those errors are, the macrophages, the eating cells, just couldn't keep up with the leukemia. So this is a clue. Well, you don't want to just make a discovery and take it this far and then throw it over the intellectual property transom and hope somebody does it. I had that lesson. That was the problem I had with a large company that wouldn't carry forward the breast cancer patient cancer-free stem cell transplants. So through the good offices of Proposition 71, which we put forward before the people of California in 2004, the state committed with a 59% to 41% vote to create an agency that could fund discoveries like this through clinical trials without having to sell it to pharma, without having to start a new company, where friends here, even in the first row, will say what you do every day if you run a company is you wonder where the next dollar is going to come from. And you spend all of your time thinking about that rather than moving the discovery forward to people. So the state could fund, and many parts of it, the state could even get a return from successful therapies that come from it. So we needed to do that. We did that. And what you're looking at now is a clinical trial just reported this year where patients who had refractory acute myelogenous leukemia, untreatable. And we treated it with both 5F9, which was the name of our blocking antibody, and a drug that helps lower the dose of cells and induces eat me signals in a lot of cells. And what you're seeing on the right is leukemia growth. It goes up in the patient if it's growing. It goes down if it isn't growing. And in both acute myelogenous leukemia and that disease called myelodysplastic syndrome, so far they're all going down, or virtually all going down. Now, that doesn't mean tomorrow or 3 years from now that it necessarily will be a drug, but I can tell you that we, the scientists who made the discovery, at a not-for-profit setting at Stanford, directed the progress of this to make the drug, the antibody, to be safe and effective potentially. We found that the don't eat me signal was on every human cancer. And all those on the right were cooperations with surgeons who allowed us to obtain, with the patient's permission, part of the tumor. So we could start looking for transplanting a brain cancer into the brain of a mouse. That's actually glioblastoma. The mouse has to be immune deficient, or else their T cells would reject it. And on the right are those that we treated when the tumor was small with a blocking antibody that is now in therapies. And all of those we tested, but they work best on small tumors, not massively big tumors. We know, and you know, how to get most of the cancer cells gone from a woman with metastatic breast cancer. So we know what we want to try as the follow-up for the other 2/3 of the patients. That molecule is the dominant don't eat me signal, and I write, because I keep forgetting, that because I co-founded the company, am a director, have stock in it, you might think I'm biased. You might have a good reason to think I'm biased, but this is the data that comes through from independent groups testing the therapy with the FDA oversight. Now, this caused us to wonder if this system of eating macrophages might explain how some antibodies work to get rid of other cancers. And one of the ones that we like and we studied, and I don't think we have the antibody name there, so there's an antibody called rituximab, which sees a molecule on normal B cells and all B-cell lymphomas. And by itself, it induces long-term remissions in a lot of people. The docs I talk to say, yeah, well, because that's how it activates a particular kind of killer cell. But we decided we were going to transplant those lymphomas directly from the patient into a mouse that lacked all killer cells, T cells, natural killer cells. All they have is macrophages to fight it. When we did that, on the upper left, the tumor is growing in the purple. Our antibody stopped it cold but didn't get rid of it in the red. The third one is green. Rituximab alone, not quite as good as our antibody in this test. And then the combination got rid of most of the tumor. On the right is a patient with a lymphoma that had defied other treatments. Before and after, the dye you're seeing was in the blood, so the heart has it, the kidneys have it, the bladder has it. but there's no tumor that has it, that diagnostic. And when we look at these patients, about half of them show some response, and about half, maybe a little more, have no detectable disease. It only works with a combination. But that's the formula going forward. Block a don't eat me signal, add a new eat me signal, that red antibody, which binds to an eating receptor on the macrophage. I forgot to tell you that. I realized it. Okay? So you block don't eat me, you add more eat me. It worked in breast cancer so long as the breast cancer was small. So Mike Clark, now a Montana ranch owner, and friend Sharon and I began working on cancer stem cells. They found the human breast cancer stem cell for a whole subset of breast cancers. They could isolate the cancer stem cell pure. They could put it in the mammary gland of an immune deficient mouse, and it grew. So we did brain to brain, blood to blood, breast to breast. So we're not inventing all the wheels. Tumor grew. If you gave them the antibody while the tumor was still small, tumor didn't grow. So can we find a second antibody to go with it? So this is an antibody called Herceptin. It's got another name. On its own, it gives remissions but cures nobody. But together with anti-47 in the first experimental test, we have cures in mice that have the human tumor. So we'll look for that. And in the meantime, we developed a general method to look for other don't eat me signals. There are 4 that dominate now. And for every cancer, this is what personalized medicine will turn into for cancer. You'll get a sample of the tumor. You'll test your antibodies to each of the 4. You'll know on any single cell what fraction of the antibodies are binding to 47, what fraction to PD-L1, what fraction to MHC1 beta-2 microglobulin, sorry about the language, and so on. And then you'll say this patient needs a combination of these 2 or these 3 antibodies. And that's what's being in test. Because this now leads us to have the chance to treat all of those cancers that we were there. We know we have to debulk the cancer by high-dose chemotherapy or surgery or radiation and follow with it. Then came a big surprise. Just pay attention to the bolded samples. We wondered if the eat me and don't eat me signals were on any other disease cells. So Gerlinda Vernig and I found in humans with pulmonary fibrosis, an always lethal disease, where the fibroblasts that are a small proportion of cells in the lung take over the lung, close off all the air spaces. You're on oxygen, and after that, you have really almost no chance. I mentioned the atherosclerosis thing. We now have the mechanism. In the blood vessel wall of the coronary artery, or the carotid artery, or the aorta, a single cell undergoes a change to make a clone of smooth muscle cells. And they call in macrophages to eat them, but they can't because the smooth muscle cells in the lesion itself that will cause a heart attack expresses CD47 and the eat me signal. So we made mice that would get atherosclerosis. We put them on a high-fat diet, my diet. And all of them were dead in 2 months. But if we treated them throughout the period with a blocking antibody, CD47, it barely made a disease at all. Now, that doesn't mean tomorrow we're going to have it. But what I'm telling you is there are rogue cells in the body, probably starting out like that first blood-forming stem cell that expanded in competition with the other stem cells that cause other diseases. You wouldn't even think there would be a relationship to cancer. But here we have it in pulmonary fibrosis, scleroderma, liver fibrosis, fatty liver fibrosis. We know that it happens in there. We've tested the human samples, made a mouse model of it. Atherosclerosis, we're not quite sure how it will be tested, but we know that's a target. Peritoneal adhesions, some of you have had surgeries or infections of the GI tract or the reproductive tract, and you get adhesions. The adhesions are fibroblasts starting to glom onto and close off all the organs. We just published, that's the Tsai one, that they're an aberrant rogue cell that returns to a gene expression program of a more aggressive precursor, and they put on 47, and we can wait until that lesion develops and clear it out. If you have peritoneal adhesions and you go to your surgeon and you say, this is horrible, you know, I'm constipated all the time and all these things happening, they will eventually try surgically to remove the adhesions. And that just starts another round of those people who are, for one reason or another, prone to develop adhesions. We found that we could cure that in the mouse model with our blocking antibodies. And recently, with Kim Hazen crew, we've been studying persistent infections. And we have a whole set of them that we're looking at carefully to see if we can change how we treat infectious disease. And you should ask Kim to come over and give a talk on it. This is the atherosclerosis in the paper we published, so it actually got published. This is the peritoneal adhesions. They look ugly. This is pulmonary fibrosis. On the left is how the disease looks. On the top is a real lung with lots of airspace. They're filled scar tissue. But if we use the right combination of antibodies, after it looks like on the left, we can take it back. So I don't want to be a snake oil salesman, but I do want to let you know that all of this came from the ability to study stem cells and follow where they took us. And even though companies like to say, we're an oncology company, we're an infectious disease company, we're an anti-inflammation, the body doesn't work like that. It doesn't know the concentration of capital for those companies. It wants to know what's going on that makes these rogue cells. And we were lucky enough to find a major step for rogue cells. Now, I'm going to finish with one part here because it was the reason that I started doing this science. When I was in high school in Great Falls, Montana, working with Ernst Eichwald, a paper came out in 1953 that you could put blood-forming or immune cells from an adult mouse into a completely different mouse while it's a fetus. And it would grow up accepting as self both cells from the donor and cells from the host. They called that immunological tolerance. And I actually did it in high school. Now I probably wouldn't even be able to work with mice because they say, oh, you got to be over 18 in California. Because they're getting in the way because we know better. You know, 17, not going to work. So we know that when we do a bone marrow transplant from a donor to a recipient, we give high-dose radiation or chemo to the recipient to kill off their cancer or to get rid of diseased blood cells like those that cause sickle cell anemia. We want to get rid of that. But it's a high dose of chemotherapy or radiation to do it. If you transplant in all of the cells in bone marrow, way back in that early slide where there were T cells there, if you take raw dead skin and put it on my body, it's my T cells that will kill her skin and reject it. So my T cells put into her body, and she's immunosuppressed by radiation, will go to her skin, some, and start killing the skin, and others to the lung, and others to the liver. So you end up with all of the organs in the body undergoing an immune attack from the transplant. And not the stem cells in the transplant, but the T cells. They're built to do that. We found many years ago that if you purify stem cells, there's no T cells. You can do any donor-host combination you want. There's no graft versus host disease. Now, before you think that I'm against all bone marrow transplantation, some of those T cells will fight off a lymphoma or a leukemia. It's only some. So you get the whole body disease, you're in an isolator unit for weeks or months, your bill is $500,000 to $1 million at least, but you're alive. And you come to a celebration every year that art of the physicians worked. But if you don't need to kill off the cancer, if you need to get a new blood-forming or immune system, those T cells don't help you at all. So to make a long story short, we found in juvenile diabetes, the T cells are the genetically defective cells that kill the insulin-producing cells. That's what that slide shows. And if you transplant bone marrow or purified blood-forming cells from the diabetic person back into himself or herself after radiation, it buys you a month. That's been published now for 18 years, and the bone marrow transplant community hasn't gone beyond that yet. We did pure stem cells from them, and they still get the diabetes, but it delayed for 2 months. But if we take, this is the important part, blood-forming stem cells from a donor that genetically didn't get type 1 diabetes, didn't make T cells that killed off their insulin-producing cells, we cure them. We cure those before they have diabetes, if we clear them out before all of the insulin is gone, or we co-transplant from the same donor insulin-producing cells and blood-forming stem cells, and it cures them for life. Published that, 2001. No single group, certainly not the large pharma that bought it from us, did it. But we were worried when we wanted to carry this forward to children with diabetes, to kids with sickle cell anemia, the true sickle cell anemia, to Mediterranean anemia, to, in fact, all autoimmune diseases. We wanted to replace that, but we couldn't put them through a next to death high-dose chemotherapy to do it, because you'd kill off probably as many people, and they could stay alive for a long time on insulin. So we decided we needed to find antibodies rather than a radiation or chemo to clear out the dangerous cells in the recipient. So we have one antibody that we found works to clear out blood-forming stem cells. That's that red one called, that language I was going to say, anti-c-KIT. We went through and found that the combination of anti-CD47 and anti-c-KIT allows us now to get successful transplants from a genetically identical donor. No radiation, no chemo, no clearing the space. We then did a test between siblings that share the most important gene for success or failure of transplants by immune rejection, but are different on all the other ones. For that, we had to add an anti-T cell antibody because T cells cause the rejection. And it worked. And we published 2 months ago, let me just go back. In the early era of bone marrow transplants, people wanted to have you have a matched donor. matched at this gene. That's a transplant gene. And you have a 25% chance that one of your siblings will match. 0 chance that your parents or kids will match. It's genetics, and I'm not going to talk about it. Just to say, that was fine when people had, like in my family, 4 sibs. I have 5 kids. Now it's 2 or 1. There's not a chance that that will work for the general populace. So we decided, Benson, George, and I, to develop a system with antibodies to clear out the dangerous cells that would work between a mother and a child, a sibling that's a half match, a child and the mother. And it works. And we put in heart transplants. That's what those little things are. Put a heart from a mouse into the ear of a mouse. It's beating. When the rejection happens, it stops beating, so it's an absolute marker of whether there's an immune rejection. It's now over 360 days, and those hearts are still beating. If the stem cell you put in came from the donor of the heart. This will be the last thing I say. And I've got a bridge to sell you if you believe that. We want to get rid of, for regenerative medicine, we want to get rid of all radiation and chemo. We want to substitute these antibodies. We want to remove their blood-forming stem cells, their T cells, and the natural killer cells that reject mother-to-child grafts. We want to be able to take a tissue stem cell like the one my wife and I transplanted into over 70 patients? 50? With spinal cord injury, with congenital diseases, with age-related macular degeneration and blindness. And we found that somebody in a business decision decided to change the immunosuppression, that they actually needed to be continuously immunosuppressed. So now we want to take a blood-forming stem cell and a brain-forming stem cell from the same donor. If we want to fix spinal cord injury or halt the progression of blindness, of macular degeneration. In the far future, and we can't oversell this because we could be wrong, we know that embryonic stem cells can make every tissue. Embryonic stem cells come from the stage of development of a fertilized egg. It takes about 3 to 4 days to develop what we call the blastocyst. It's something that will implant in the uterus. You can't do it before because it doesn't have the cells that attach to the uterus. Once it attaches to the uterus, it makes us. The cells that are in that early stage make all of the tissues. And what the scientists who study embryonic stem cells know is that you can grow them artificially and keep them at that early stage, embryonic. And because they make every tissue, we're looking for the stem cells of the tissue, and Kyle Lowe and I have found several already. So in the distant future, the reason you don't want a political or religious ban on embryonic stem cell research, or what's called induced pluripotent stem cell research, is that that will be your therapeutic chance. Antibody conditioning, stem cells, and the other tissue cell. I'm convinced it will work, but I'm not convinced how long it will take to work. So, if you think about Maurice Hilleman's career, he proved that it was possible to make vaccines to protect people, most people, for life against some of the most deadly infections. He led a company called Merck to develop that and market it, and literally saved millions of lives. But the drug company, and most drug companies, feel best if they have small molecules that they can sell every day. So the breakthrough that we're talking about here, starting with Hilleman, with not small molecules, but immunization, us with stem cells and other cell therapies, and all that comes from it, is that we shift from the paradigm of getting soil samples to find chemicals, to run through a whole bunch of cell lines and hope it kills or does what you want, and then eventually develop a drug. That's what the 20th century did beautifully. But if you stick with that, you will be stuck with 20th century medicine. And what we need to do is to move it forward with curative therapies that happen just once. The stem cell self-renews for life. Any stem cell therapy is once and done. So, in honor of Maurice Hilleman and in pleasure of coming back to MSU, I'd just like to say, this is where we are, this is where we're going, and despite turning 80 in a couple months, I ain't done yet. Thank you. So we have questions?
Sure, absolutely.
Yeah. So we have time for questions. Please sit down. Hey, wife, did you get a picture of everybody standing up? Anyway, so questions. I know it was complicated. It was a long talk, but any question from lay people, scientists, disbelievers, whatever? You're all convinced?
Can you touch on your ideas of the T-cell, the CAR-T therapy that's coming about?
The T cells that are used in CAR-T, yes. So T lymphocytes have many properties. Long life. They have a receptor that allows them to see a marker on a cell and kill it. But that system that is naturally there is so complex, it will take us decades to figure out which T cell was graft versus host and which one was graft versus leukemia. So a very inventive Israeli named Zelig Ashar said, oh, that's easy. I'll just put an antibody on top of the molecule the T cell uses to recognize foreignness and kill. And I told him, of course, that can't possibly work because a lot of those molecules are also free in the bloodstream, and it would be a radar, like interfering screen. So, of course, it worked. And it took him about 10 years or more to get people to try it in systems. And so now what they do is they take a T cell from your body of the killer type, and they insert a whole gene complex that if it binds to a target cell that has the marker, like a B cell marker, and you have lymphoma or acute lymphocytic leukemia, it will kill that cell. If you have too many, that releases the proteins called cytokines that cause inflammation, and you can die from loss of your blood pressure. So it's not yet a safe therapy. It costs $500,000 at current rates. So it's not a general therapy. But if you're somebody who got saved, it's very good therapy. Maybe we need a drink. Thanks. Oh.
How can patients find out and providers find out if there's a human trial?
I can't see you, so I can't read your lips. I'm half deaf. Say it again.
How can a person find out if there's a clinical trial for people if they're interested?
So we do in our therapies, every one of them, all the ones I've showed, we went through the humanized mouse first. So that's a mouse that's immune deficient that has human blood forming and immune organs that we can measure and follow. The next step, if the targets are the same in non-human primates as humans, you can then test it in non-human primates. And we did that extensively, and we found some unexpected toxicities and the way to get around them with the anti-47 therapy. In the clinical trials, except for a loss of red cells, and not many on the first injection, because remember the mechanism that CD47 works is to keep red cells alive and away from scavenger macrophages. If you block it, you lose some red cells. So you give a low dose the first week. The next week you can give 90 times that dose in the bloodstream and do it every week. No adverse events related to the antibodies. But that's because we were scared to death as academicians, developing something that companies usually develop, that we would have an uncovered toxicity, which would ruin everything.
If you want to find out whether one of these therapies is suitable for a patient you know or yourself, Google clinical trials. And you will find a website by the FDA with all of the approved clinical trials.
Good, thanks.
These anti-CD47 antibodies that were created, do they cross over the blood-brain barrier in order to combat these symptoms?
Terrific question. So most antibodies, most proteins, most cells can't cross into the brain because there's a barrier, astrocytic mainly. In glioblastoma, the barrier gets destroyed. So in the mice and in the humans we're about to treat, we just give it systemically. And it crosses and it covers. And there's a special kind of macrophage, eating cell, in the brain. Again, Latin, microglia, which is totally wrong. It's not a glial cell. But anyway, they do the eating of brain cancer. We've done it with glioblastoma, childhood diffuse intrinsic pontine glioma, medulloblastoma, astrocytic astrocytoma, and all of those tumors from a human put into the brain weeks to months before, keep the blood-brain barrier open and the antibody gets there. And the macrophages, microglia, eat it.
On the flip side of that, then, are we seeing autoimmune diseases in the body and in the central nervous system actually producing too many antibodies?
Yes. So in multiple sclerosis, you have T cells and antibodies against oligodendrocytes. In what my mother had, Evans syndrome, antibodies against her red cells and her platelets. In lupus, which is a pretty horrible disease, antibodies against DNA or RNA, which form complexes, get filtered on the kidney and kill the kidney, and they die of renal failure. So yes, in all of those autoimmune diseases, you do see antibodies, and you see antibodies against the relevant targets.
We'll take one more. One last question.
Hi. I'm a public scholar, and I was just wondering, what the state of undergrad research was when you were going to school, because we're focusing a lot on it right now.
I worked in Ernst Eichwald's lab every summer from the time I was 16 until my clinical years in medical school. That opportunity has been available in Great Falls ever since then. And of course, you look here, there are many labs that undergraduates are working in here. That's our most important legacy, is to let people find their talent. It isn't going to be determined by your grade point average, I promise you. So if you are lucky enough to go to Montana State, there are many labs you can find, and you'll find out if that happens to be your particular talent. It's just as important to find out it isn't. That is, you want to go home, the time is marching on, or you end up staying all night. You'll know when it's right for you.
Thank you so much.
Right.
So we want to thank Dr. Weissman for a wonderful lecture. I'm still thinking about this case about transplanting my skin to you. I don't know how much it's going to hurt. On the question of, from our Hilleman scholars, this is a great opportunity for me to remind you, Montana State University has been chosen as the host of the National Council for Undergraduate Research. We're going to be hosting the annual meeting here, March 26th through the 28th of 2020. Over 4,000 undergraduates from all over the nation will be here in our campus showcasing their undergraduate research, including students from every campus of the Montana University system and all our tribal colleges. So I hope that to see you then. And one more time, would you please join me in thanking Dr. Irving Weissman for a wonderful lecture.